Hello everyone, in this video we'll be discussing about Kelvin cycle, which is the dark phase of photosynthesis. As we all know that photosynthesis has two phases, one is the light phase and the other one is dark phase. In light phase there occurs four important things. One is the light is being trapped through light harvesting complexes. Second step and third step are related. Second step is splitting of water and the third step is releasing oxygen. And the fourth last step of light reaction is production of high energy compounds which are ATP and NADPH. Now here in this dark phase reaction discussion, we'll start with Kelvin cycle, that is the most important topic of the entire chapter. So this is a part of biosynthetic phase of photosynthesis, which is also called as dark reaction. But calling it a dark reaction, is it worth it? No, it's a misnomer that this process directly does not depend upon the light, but it surely depends upon the products of light reaction. For example, we have discussed in chemiosmotic hypothesis that the ATP and NADPH produced in that reaction, which were in stroma, is actually used in this dark phase. And if light is not available, how come the plant will synthesize ATP and NADPH? Therefore, this dark reaction would also stop after some time when the light is deprived to the plant. Therefore, it is misnomer. This reaction does not directly depends upon light, but surely depends on the product of light reaction, and the products are ATP and NADPH. Understood? Why it is a misnomer? Because this reaction can function in light also. It's not as such it needs darkness to be there. Now let's talk about the scientist, the god who gave his life to photosynthesis, and is Melvin Kelvin. Just synonymous names, Melvin Kelvin. He used radioactive carbon, that is C14 atoms, and he did his research on algal photosynthesis. Which scientist we have discussed also used green algae? The answer is T.W. Engelmann. He used the green algae Cladophora while he was splitting his white light into spectrum. He discovered the first carbon dioxide fixation product. He was the first scientist who said, wait, let me give you the entire cycle how plant is converting carbon dioxide into glucose molecule. So he first of all when he started, he found the first carbon dioxide fixation product, and then he was motivated to work the complete biosynthetic pathway of carbon dioxide fixation. And this complete biosynthetic pathway is of two types. He was surprised. One is C3 pathway and the other one is C4 pathway. And similarly, the plants performing C3 pathway are called as C3 plants, and the plants performing C4 pathway are called as C4 plants. So this dark phase of photosynthesis has divided the plants into two groups. So what is the difference? So the plants in which the first carbon dioxide fixation product formed is 3 carbon containing organic acid are called as C3 plants. Similarly, the plants in which first carbon dioxide fixation product is 4 carbon containing organic acid, it is called as C4 plants. So what is that 3 carbon containing acid? It is 3 phosphoglyceric acid. This is an intermediate of glycolysis. We have discussed this in under the heading of glycolysis also. So this is the first carbon dioxide fixation product in C3 plants. Whereas in C4 plants, the first 4 carbon containing fixation product of carbon dioxide was oxaloacetic acid, OAA. Now let's see, who is the acceptor of carbon dioxide molecule? What do we mean by acceptor? We mean to say that which molecule will first combine with carbon dioxide to form either a 3 carbon containing acid or a 4 carbon containing acid. Scientists thought, like, see, carbon dioxide is a single carbon containing molecule and the product formed is 3 carbon containing. Therefore the acceptor should be 2 carbon containing, but to their surprise the acceptor was 5 carbon containing. 5 carbon containing ketose sugar. As we will discuss in biomolecules chapter, that sugars are of two types. One is aldose, which have aldehyde group in them, and one is ketose sugar, which has ketone group in them. So therefore the primary acceptor of carbon dioxide was a ketose sugar, that too 5 carbon containing. Then scientists said, how come a single carbon containing molecule fuse with a 5 carbon containing molecule and give a 3 carbon containing product. So scientists said, Duffer, can't it be like 1 carbon containing substrate combined with 5 carbon containing substrate to form 2 times a 3 carbon containing product? And they were like, yeah, this could be possible. They said this is only possible just because the primary acceptor is a 5 carbon containing ketose sugar whose name is ribulose bisphosphate. In short, that is written as RUBP, ribulose bisphosphate. So this is the primary acceptor of carbon dioxide. Now let's talk about the Kelvin cycle. Kelvin cycle occurs in all plants, be it C3 or C4. What do we mean by Kelvin cycle? Kelvin cycle means fixing carbon dioxide into glucose, and the whole path is called as Kelvin cycle. So all plants fix carbon dioxide into glucose, be it C3 plants or C4 plants. So it occurs in all photosynthetic plants, be it C3 or C4. There are 3 stages of this cycle, Kelvin cycle. First stage is carboxylation, second stage is reduction, and the third stage is regeneration. Let us discuss each of these stages in detail as per NCERT. Don't panic. So everything which I am discussing is mentioned in NCERT textbooks. So the first reaction, carboxylation reaction, is actually the formation of first carbon dioxide fixation product. So here we are fixing carbon dioxide into stable organic intermediates. When carbon dioxide is utilized for the carboxylation of the acceptor molecule, we know that acceptor is RUBP, a 5 carbon containing ketose sugar, ribulose bisphosphate. It results in the formation of 2 molecules of the product, that is 3 phosphoglyceric acid. We know that phosphoglyceric acid is 3 carbon containing organic acid. Therefore we need to balance the equation. Therefore 2 molecules of this acid is produced. This is the most crucial step of Kelvin cycle, and this is catalyzed by an enzyme called as RUBP carboxylase oxygenase enzyme. So it has a part which is called as oxygenase enzyme. We have not taken this P in the short form. Directly ribulose bisphosphate carboxylase oxygenase. Therefore it is also called as Rubisco RUBP carboxylase oxygenase. Friends, this enzyme is made up of protein. As we know, most of the enzymes are proteinaceous in nature except some enzymes which are made up of RNA. So this is the most abundant protein on this biosphere, on this planet Earth. The most abundant protein of animals is collagen. This is mentioned in biomolecules chapter of NCERT. So you need to remember that most abundant protein of animals is collagen, and of entire Earth is this enzyme Rubisco, because every plant will do photosynthesis, every plant will have Kelvin cycle. Kelvin cycle first step is carboxylation, which is catalyzed by this enzyme. So this enzyme is present in everything which is green in color, which has chlorophyll. Now this is all about the first stage of Kelvin cycle, that is carboxylation. Carbon dioxide is received by an acceptor molecule which is 5 carbon containing ketose sugar called as RUBP, ribulose bisphosphate. And when this carbon dioxide fuses with RUBP, it leads to the formation of 2 molecules of 3 carbon containing organic acid, which is called as phosphoglyceric acid. Understood? And this reaction is catalyzed by an enzyme ribulose bisphosphate carboxylase oxygenase. Why it is called as carboxylase oxygenase? Because this enzyme has a low character, that it does not take its own stand. It says that if oxygen will be more, I am oxygenase. If carbon dioxide will be more, I am carboxylase. So it has no stand of its own. It depends upon the surrounding concentration of gases. Therefore it is called as carboxylase oxygenase at the same time. Now coming to the second step of Kelvin cycle, which is reduction. And this is actually the reactions which lead to the formation of glucose molecule. So these reduction reactions are actually series of reaction which results in formation of glucose. Carboxylation was the first step of this cycle, where we fix carbon dioxide into first stable organic acid, that is 3-phosphoglyceric acid. So in this process of reduction, energy is used. As we were already talking that in dark reaction, we need the the products of light reaction, that were ATP and NADPH. So here in the second step of Calvin cycle, we are using those products. We use 2 ATP for phosphorylation and 2 NADPH for reduction in the second stage. 2 to 2, you could remember the second stage, we use 2 ATP, 2 NADPH for reduction. Now friends, let me bring it to your attention that for synthesis of one glucose molecule, we require 6 carbon atom. Therefore 6 carbon dioxide molecules should be fixed. Therefore 6 turns of this cycle will be taking place to produce one glucose molecule. Then how much ATP would be required in the second step? If we see 6 into 2, that is 12 ATP, 6 into 2, that is 12 NADPH. 2 means these are 2 ATP and 2 NADPH of one cycle, and we are talking about 6 cycles, therefore I have multiplied by 6. So just for the second stage, we require 12 ATP and 12 NADPH for synthesis of one glucose molecule. Now let's see about the last stage. Last stage is of regeneration. We want to accept the carbon dioxide more, therefore we need that primary acceptor more and more. Therefore we generate the primary acceptor, which is a 5 carbon keto sugar named RUBP, ribulose bisphosphate, and this regeneration step takes 1 ATP, and therefore for fixation of carbon dioxide into glucose we need 6 cycles. Therefore 6 into 1 ATP, that is 6 ATP will be required for regeneration step. In total we require 12 plus 6, that is 18 ATP and 12 NADPH for the formation of glucose by plants. Let me discuss the cycle mentioned in NCERT textbook with you. So here we start the journey of Calvin cycle when atmospheric carbon dioxide fuses with the 5 carbon containing keto sugar, which is the primary carbon dioxide acceptor named RUBP, ribulose bisphosphate. It could be questioned that where are the phosphate molecules attached? These are attached on the first and fifth position. If someone wants to ask a trick question, it is ribulose 1,5 bisphosphate, just because it is mentioned in NCERT. So when it combines with carbon dioxide, the first step is carboxylation, and this is catalyzed by an enzyme called as Rubisco. You need to write this enzyme similarly as I am writing: R capital, U small, then B capital, I and S small, then C and O both capital. So R, B, C, O are capital letters in this name of this enzyme. So Rubisco will catalyze this first step, that is carboxylation, and will lead to the formation of 2 molecules of 3 carbon containing organic acid called as 3-phosphoglyceric acid. Now the second step, that is a series of steps which we are calling as reduction, will take place. And in reduction, for each molecule of carbon dioxide we require 2 ATP and 2 NADPH for this reaction. Then will lead to formation of a triose phosphate, which we have dealt in glycolysis also. I'll be telling you this again. When we multiply this reaction, this whole cycle 6 times, it leads to the formation of glucose, which would be stored as starch. And now coming to the last part of Calvin cycle, that is regeneration of the first primary acceptor, that is RUBP, and in that 1 ATP is required in the regeneration process. So total for 1 carbon dioxide we require 2 molecules of ATP plus 1, 3 molecules of ATP and 2 molecules of NADPH for fixing 1 carbon dioxide molecule. So 1 carbon dioxide, 2 plus 1. 2 molecules were used in reduction and 1 molecule was used in regeneration purpose of ATP, and 2 molecules of NADPH were used in reduction process. Now let's come to that glycolysis part. The story is simple. Actually what is happening in plants is similar to gluconeogenesis taking place inside the liver and the kidney of humans. We humans are also capable of synthesizing glucose from different substrates. Our substrates could be glycogen, it could be lactic acid, it could be amino acids. So this occurs when our glucose stores get depleted. Then body says that I don't have glucose, convert anything into glucose so that I could survive. So this process is called as gluconeogenesis, and this diagram which you are seeing is actually from the NCERT showing glycolysis. So if we move downwards, it is called as glycolysis. But if we trace it back towards glucose, it is called as gluconeogenesis, that is forming glucose from new substrates. Now in photosynthesis also it is similar to gluconeogenesis. What is happening is when carbon dioxide is fusing with the primary carbon dioxide acceptor, which is a 5 carbon containing ketose sugar, it is forming two molecules of 3-phosphoglyceric acid, which is similar to that of substrate of glycolysis. Now what will happen when we move up? We know that NCERT has not used the markers of 2 ATP molecules here, also 2 NADPH molecules here. So here we can see that 2 ATP molecules were formed, but since we are going upwards, uphills, 2 ATP molecules will be used, similar to that in reduction process of photosynthesis. Again here 2 molecules of NADPH are formed in glycolysis, but in photosynthesis, since we are going against the flow, 2 molecules of NADPH are used, and that led to the formation of triose phosphate. Let me just connect this to you. See, this phosphoglyceric acid is going through the process of reduction, where 2 molecules of ATP and 2 molecules of NADPH are used to form triose phosphate. This is actually the reversal of glycolysis. When 3-phosphoglyceric acid, we are moving towards triose phosphate, and now the journey would change. Actually, most of the carbon dioxide fixed would leave this cycle for regeneration of RUBP, and only some part would lead to the formation of glucose. And for your kind information, these 2 ATP requiring steps are irreversible steps. Therefore we require fructose 1,6 bisphosphatase and glucose 6 phosphatase enzymes, which do not lead to the formation of ATP. Understood? So overall, when we move from this phosphoglyceric acid to glucose, there occurs only expenditure of 2 molecules of ATP and 2 molecules of NADPH molecules. I hope I am able to connect this in your mind, that how glycolysis is actually related to photosynthesis. Plants are not doing something just out of the box, they are doing similar process which we do in gluconeogenesis, but they can do this in the presence of light using carbon dioxide, but we require glycogen, which itself is made up of glucose. So plants are valuable to us just because they can convert carbon dioxide into glucose. We cannot convert that as such. We need certain substances containing glucose like glycogen, or say lactic acid or other substances, for converting them into glucose. So this was the complete discussion of Kelvin cycle. I hope you like this detailed discussion and how I related this cycle, Kelvin cycle, with glycolysis. If you like this, do mention in the comment, and for more such relevant content, do check my channel also. I'll upload this whole notes on my Telegram channel. Do visit Telegram. Thank you for watching this video till the end.